Everybody say hi.
Hi.
And now you can tell your mom that you're on YouTube. I don't know if this is going to work or not. So when you know, in Moodle, I'm not sure if you looked at any of the videos yet. I think I am better in person than I am on the video. But there's, you know, a ton of lectures on there. Plus there's like, I think like videos that I've watched and stuff. And like, so if you go to Dr. Galuski's lecture video channel, there's all kinds of weird stuff on there that I'm like, oh geez, I'm not sure you should. And then of course, you know, people can put comments in, and occasionally there'll be a like for the video. Oh, I like your video. One of the things that I'm really keen on watching is when we get into virology, there is some kind of googly type spider surveillance system that I think just is actually taking words that I say and, you know, it's like, you know, generating subtitles and things. It knows that this is a lecture on viruses, and there are like 4 or 5 comments from people that are not part of the class that saying, I had this virus and Dr. So-and-so cured me of it by, you know, these various treatments. Go to this website. I'm like, dude, that is just really inappropriate. You don't even know who the hell I am. And there's no way you watch the video. And yeah, but so we look for that. So in one sense, I have not seen that yet with Molecular Cell, although I haven't done those lectures entirely online like that. But we ought to throw out key terms and see if the bot picks it up. So today we're going to be talking about viruses and the mechanism by which environmental conditions affect viruses. And I will be bringing forward my theory that viruses are in fact aliens that have invaded us and are trying to subtly shift our genetics so they can lay their young inside us. This I believe. I have said, I have spoken. Now, if I get comments on that, that's going to be awesome.
That's like a good topic for a TED Talk.
Might be, tell you. All right. Well, one final point. One of the things that I play with for student visit day, which we're not even doing anymore, I have like a whole discussion of making recombinant humans where we can add DNA and thinking about Clone Wars and, you know, the Camino cloners, if you know your Star Wars. And what are some of the genes that I think would be interesting to put into my super soldier? And there are a variety of things that would be interesting to do. One would be to provide people with the ability to biosynthesize their own vitamin C, which is kind of like a pet project I've been playing around with, not to actually put it into humans, but to looking at what organisms, what different creatures can biosynthesize vitamin C. If you can make vitamin C, it would probably have a phenomenal effect on your ability to heal from wounds, and it might actually cause a life extension. We can't produce our own vitamin C. There are genes that would potentially modifications that might allow you to see a little bit into the infrared spectrum, so you could have like some cool night vision. A whole range of things. We'll see if that gets out there too. Trying to crank up your immune system is a bit of a trick, because a lot of stuff that you can do ends up actually just is a shortcut to cancer, and that's one of the things like trying to create longevity. If you want to try and live forever, that's called tumors, and no, we don't want to do that. Otherwise, the ability to survive longer without oxygen would be a cute trick, which brings us to this point here. So when we start thinking about animals, all animals are consuming carbon for electrons, for energy, for carbon. It's a one-stop shop. With bacteria, though, as we were mentioning, you've got different bacteria strains and species that actually can extract electrons from minerals. They can get energy from different salts, and they're getting carbon from carbon dioxide, And they are way, way different than we are, and their metabolism is really different. Then we start talking about the oxygen possibilities, and we've got then a couple of different possibilities. The obligate aerobes, as we are, they have to have O2 for respiration to make ATP. Microaerophiles, they are able to operate with small amounts of oxygen. Facultative anaerobes prefer oxygen, and they can live without it. Yeast are a really good example. And most like all the bacteria that we were working with yesterday are going to be under the category of facultative anaerobes. They don't grow very fast without oxygen, but they will grow. And then we have some oddballs that we actually don't grow here in the building. Obligate anaerobes are guys that are killed by oxygen. You can imagine that incubator has got a great big door that you open up. There is plenty of oxygen exchange. That petri dish lid is not sealing itself up, and certainly there is an atmosphere in that little space, and they're operating. If you do a stab in a deep, you might begin to generate an anaerobic environment way down at the bottom of that tube, as the auger kind of closes up, but it's not particularly anaerobic. To grow anaerobic bacteria, you have to have often a CO2 incubator, and it's a whole different way of operating with them. I've seen things where you can get like this long plastic tube that you stack your petri dishes in, and then there are a variety of different chemicals that you can kind of crack open and dump in this little packet that'll scavenge up the oxygen. Or another trick is you put it into like a paint can or some sort of metal container, and you put a candle in there, light the candle, seal the lid, and as the candle burns, it consumes the oxygen. It doesn't go down to a zero oxygen environment, but it definitely goes down to a much lower oxygen environment, and you can grow anaerobic bacteria that way. But I've never grown any kind of anaerobic bacteria. And there's some pretty nasty ones too, so no, I don't want to grow anaerobic bacteria. But that was the way we left that kind of environmental conditions. The other environmental condition that we need to throw in is pH.
Nursing facilities, so we've got our nursing lab in here. We've also got on the roof of our observatory, which is
So, most bacteria are nucleophiles, about 6-8, and if you're between pH 6 and pH 8. And most of the pathogenic bacteria are going to be around here because that's what most of our body is. I mean, in your gut specifically or your stomach specifically, you'd be pretty low. Acidophiles are working at a pH between like 5 and 6. They don't go real low. Extreme acidophiles, being able to live below pH 4 is not easy. And it's also not easy to find places that have pHs that are that low. But like mine tailings and things can generate sulfuric acid and hydrochloric acid and stuff. And you can end up with some bacteria that actually can live in really, really harsh environments. Just interestingly, there aren't very many bacteria that operate in high pH, mostly because there's just not a whole lot of high pH environments. But like the Great Salt Lake, stuff like that, has a pH probably of around 9 or so. But it's hard to find places that are that much more alkaline. If you find them, there are going to be bacteria there. So acidophiles are actually pretty cool in that there are a variety of bacteria that are used for fermentation and or for fermenting foods. Things like yogurt and sauerkraut, the ability to make vinegar, these are bacterial products, and they're bacteria that operate at low pH or below between 5 and 6. And so one of the things you can do is culture these specific bacteria. To make yogurt and making sauerkraut is actually fairly easy, where you just take cabbage, slice it up, put salt into it. Most of the bacteria that are around 6-8, between 6 and 8, are not going to grow in these salty conditions, Additionally, the salt begins to extract some of the acids in the cabbage, keeping the pH fairly low, and you generate culture conditions that are perfect for these benign acidophilus and lactobacilli, and they actually are producing their own different vinegar and organic acids, which is kind of a cute trick. So that's another one set of the conditions. Again, for pathogenic guys, it's pretty much the neutrophils. Another thing, just as this is a micro class and discussing different kinds of bacteria, not just pathogenic ones, there are some guys that live under really extreme pressure conditions. So you got guys that live very deep underwater, and the pressure is quite intense. And there are a number of bacteria that have been found at extreme depth. The Challenger was a deep diving submarine. And why? We don't know. As we say, it's also medically irrelevant. But it is actually very cool, these different kinds of extremophiles. Now, something that's not irrelevant is hopefully a term that you know from A&P or from chemistry or life on the street, the idea of osmolarity. If I mention osmotic pressure, do people go, what the hell is that? Or does everybody go, yeah, I've heard of osmosis? We mentioned a little bit about osmosis as the diffusion of water across a semi-permeable membrane. And what this means is water is moving towards areas of higher salt. And what this means is the environment in which you put a cell is going to influence whether water is moving in or water is moving out. So if you take red blood cells and put them into pure water, an IV is typically 0.9% salt. So you never start an IV in pure water. Don't do it. Red blood cells that are in pure water, the osmotic concentration inside the cell is greater. Water starts moving in, and the cell will very quickly pop. Now, plant cells and most bacteria cells have a really good cell wall. And so they actually are ready for water to start moving in, and they pressurize themselves up. Now, if you've got bacteria in water, they start to swell up, and they should be able to kind of handle it, but it's not really their ideal growth conditions. So we try to grow bacteria in a moderate amount of salt. And this is the standard bacteria that we are growing in the lab. There are some bacteria that do require very high salt concentrations. For most pathogenic bacteria, isotonic conditions in which the salt concentration is the same inside the cell and outside the cell. And as I said, like for us and for most organisms, about 0.9% salt is the sweet spot. Then we can have conditions that are hypertonic, and these are all relative terms. Tonic being salt, hyper being high. In a hypertonic situation, we have the salt is way higher outside versus the inside. Water always follows salt. And so a hypertonic solution is going to cause the cell to lose water. Water starts moving out. The opposite, a hypotonic solution, hypo being below, like a hypodermic needle. Ain't nobody going to be using a hypodermic needle. That would be walking up to somebody and squirting it in their face. No, you stick it below their skin in a hypodermic. And a hypotonic, the concentrations of the salt are higher inside than outside. Water is following salt, so the water is moving in, and this is where you risk the cell popping. For bacteria, because they have a cell wall, putting them in water is not a deal breaker. That is not going to kill them too fast. But if you put them into a solution that's got a really high salt concentration, the water starts moving out, and that will start to screw them up. So if your media has got like a ton of salt or a ton of sugar, that will actually probably inhibit bacterial growth. It's not really a standard sterilization technique, but in terms of preservation and as a preservative, things like jerky or dehydrated food, they work, or in fact salting food, as a bacterial inhibitor, because that high salt means that the bacteria are not going to be able to grow, they lose too much water. And so if you want to actually consume it, you probably have to wash it a bit to remove some of the salt. So that's another one of the environmental conditions that is part of the whole growth system. And these are sort of the ways we think about the differences and the challenges. In a hypotonic solution, water is moving in. The cell wall keeps the cell from popping. And like I said, if you take your red blood cells and put them into a hypotonic solution, they lyse. Not going to be good. But if you take these bacteria or a plant cell, put them into a hypotonic solution, they swell up, and they're good. Now, interesting exception here is that penicillin is blocking the production of the cell wall. So now you got bacteria that don't have a particularly good cell wall, and when they're exposed to a hypotonic solution, they'll pop open. So that's one of the ways that penicillin and the different kinds of cillins and cell wall inhibitors are working, is they cause bacteria to now have the possibility of lysing. With an isotonic solution, you have the overall pressure is the same. You don't really need a cell wall, and for you and I, this allows our cells some flexibility to move around. So the kidneys are, in fact, responsible for regulating salts in our bodies, and we actually depend on our kidneys pretty seriously to keep our salt concentration pretty tight at around 0.9%. If your salt concentration goes even 1% up or down, you're going to be in real serious problems. You're probably going to go into cardiac arrest. So keeping your salt concentration really, really close is important. And that's one of the problems with dehydration. Is if you start dumping a bunch of water, your salt concentration goes up, and you can screw yourself up pretty seriously. A hypertonic solution, water is rushing out. The cell is actually going to end up shrinking. There are some cell bacteria that actually can put more salt into their cytoplasm, but it doesn't work very well for most guys. These organisms are known as halophiles. They actually can live in real salty environments. Your skin, when you're sweating, depending on how you're sweating, it can have a fairly high salt environment. And so there are some bacteria then on the skin that are able to actually interact with that and take advantage of that. All right. So when we think about our skin, this is like your first line of defense for bacterial infections, for sure. The viruses, you inhale them, and bacteria, you could inhale them, but that'd be a bad thing. We're often going to interact with a lot of bacteria in the world being blocked by our skin. And if we think about this, the skin is reasonably dry. That is going to help dehydrate bacteria. They don't get a lot of moisture that way. It's actually reasonably acidic, the surface of your skin is. There are fatty acids and oils that are serving to dissolve membrane, and so it's serving as a defense mechanism, not something you need to be concerned about in terms of removing the oil from your skin. Your skin can be fairly salty. There are even enzymes like lysozyme, which is able to dissolve the cell wall, make the cells that much more sensitive to osmotic pressure. And this is found in tears and a variety of other secretions. By digesting the bacterial cell wall, it's going to cause the cell to pop open. We are, maintaining a somewhat inhospitable environment for bacteria. What it means is, on your skin surface, you can get topical bacterial infections. They're mostly fairly easy to treat, and they don't happen all that much. We've got bacteria that are evolved to live on us. So you pretty much are covered in Staph aureus. And one of the things I think about is it's important we think about our bodies as bacterial parking lots, and you want to make sure that your parking lot is filled with non-pathogenic bacteria. And so things like antibiotics and various kinds of other stressors, potentially excessive use of antibacterial soaps, can strip your skin of the non-pathogenic bacteria and provide parking spots for pathogenic bacteria. So while we're washing our hands all the time, excessive use of antibacterial soaps, particularly on the rest of your body, maybe not the best thing for you. All right, so that's the external environment. Now let's think about the kinds of foods we need, the different medias. So Wednesday, we were using one media, that is tryptic soy agar, or tryptic soy broth. It is what's referred to as a rich media in the biz, in that it's got pretty much all the stuff you need for bacterial growth. It's got a fair amount of protein in the form of a hydrolyzed cow parts. It's got enough fats and lipids. It's got a lot of carbohydrate. It's got also nucleic acid monomers. So we have all the components we need for making more DNA. And it has pretty much everything that you need. So the macronutrients, carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus, these are all things we find in these biopolymers. Then we've got micronutrients, or other kinds of salts and things like iron, potassium, magnesium, calcium. These are required for various kinds of cofactors and interactions with proteins. Then you've got real micronutrients, things that are involved in like trace amounts that are probably present just in the water, things like selenium and stuff. Although we need teeny little amounts of that, and it's pretty hard to actually generate a diet that doesn't have them. So they're not really all that well studied or characterized. But a rich media is all we need for our bacteria to start growing. So we can modify the media in different ways and have different kinds of selective media. So the micronutrients, these are things, as I said, cobalt, copper, manganese. They're used in such small amounts that they're never typically added to it if you're using any kind of developed material like a broth of some kind. It'll have it in there. Let's see. So other growth factors, and these are sometimes complex organic molecules. And as we start getting into metabolism, NAD is an electron acceptor. Vitamin K. Sometimes there are amino acid modifications that are required. We can't biosynthesize 7 of the 20 amino acids, and those are referred to as essential amino acids. Most bacteria can biosynthesize all 20. Some can't, but most can. But even still, there are amino acid modifications that are often required, and sometimes the bacteria can't do that. This, from an animal perspective, there's a modification of the amino acid cysteine into a slightly different structure that's called taurine. And taurine is required for a functional protein in the eye. And we and dogs can biosynthesize taurine from cysteine. Cats cannot biosynthesize taurine. And consequently, they have to get taurine from meat, from animal sources. So you can be a vegetarian, your dog can be a vegetarian, but you cannot make a vegetarian cat, at least not a very healthy vegetarian cat. And taurine was discovered as a modified amino acid back in the 80s, and they actually were able to recognize there's a specific kitty cat blindness that was associated with taurine deficiency. And now if you go to your cat food, you'll see a little cat eyeball icon, and it says, taurine enriched, or like it always was. As long as you have meat in there, you're good. One of the things it means is that sometimes it can be difficult to grow some of these weird pathogens in the lab because they need to have something like blood agar, which we haven't made in a while, but we have the stuff to do that, where you have blood protein and a blood extract, and you mix that with the other media, and that allows for some vampire-like bacteria to grow. So what we've been using is complex media. And this is all the necessary nutrients. And it's generally a bit of a black box so that you don't really know exactly how much of the different ingredients are in there, because you're taking yeast extract, which is essentially a product of the brewing industry, where they have gigantic Bush brewery with huge amounts of yeast at the end of their brew run. They'll take it and crush it up and blow it into a fine powder. It's going to have all the different kinds of proteins and nucleic acids, all sorts of stuff, and you sell that as yeast extract. You can buy that as nutritional yeast at your local GNC. Casein hydrolysate, which is taking milk and breaking it apart, the milk protein. There is all kinds of what are called bactotryptone and bactopeptone, which is tryptic or peptic, these are proteases, digests of animal parts, mostly cow. And again, you're getting all the different amino acids and stuff. You don't know exactly what you got in there, and for most culturing, it's okay. You can get and make stuff that we think of as enriched media, in which you add other stuff. You can add glucose as a sugar solution and really get a lot more bacteria growth. And then you can have things like blood for organisms that require very specific compounds that are found in that blood. And now, it seems most of the time that blood is blood. You can get cow blood. I've never seen a recipe that actually requires human blood. That would be a creepy-ass vampire bacterium. But just like the way all our vampires these days are able to go hunting and take blood from a deer or from who knows what sort of farm animals, they don't have to prey on humans. The same thing with bacteria. Let's see, defined media is something that is used in which you have to really carefully control the environment that your bacteria are growing in. And it's not typically used. You have to provide all the essential amino acids, all the essential nutrients, and the bacteria typically grow a lot slower. Selective media is, as the name implies, is able to select for a kind of bacteria over another. And a standard way to generate selective media is to change the salt. There's a medium called mannitol salt agar that will allow staph bacteria to grow over other bacteria. And so you can select for different kinds of bacteria rather than having to do a streak or a spread to get individual bacteria. You can grow them on selective media, and whole classes of bacteria will not grow, and very specific ones will. Differential media is a kind of media that allows for the uptake of often a dye, and it will allow you to see one bacterial strain over another one. Other things might be you have starch in the agar, and there are certain bacteria that can break down the starch, and then when you stain the plate, you'll see a little halo around those bacteria where they pull the starch out. You can actually have then sort of combinations. MSA is a media that we actually use occasionally, growing the salt-tolerant bacteria, and you got a dye that is distinguishing between different staph species. So you can distinguish Staph aureus from Staph epidermidis. EMB is another media that we use, and this only will allow gram-negative to grow, and you still have a dye that will distinguish between these different kinds of species. So if you're trying to identify species, this is a useful thing to be able to do. All right. So this is the material for the first exam. And what I would like for you to do, I will on Monday, or yeah, probably let's make it Monday. I'll do a Moodle dump of kind of my lecture notes from the different chapters, and you can use that to compare with your notes. But certainly building lists of, an obvious thing would be, drop and give me 3 things that are found in prokaryotes that are not found in eukaryotes, or 6 things that bacteria and eukaryotes have in common. That kind of stuff is useful. Being able to describe and differentiate between the different macromolecules, what are proteins versus carbohydrates versus lipids versus nucleic acids, and then a variety of different terms we've been using. That's what we'll be looking at. So we haven't really gotten into too many bacteria lists of what bacteria cause what disease yet. Really not anything like that. This is a good bit more general. And that's what I have to say about that. So I'm gonna kill this.